The word organelles means small organs. So these are small organs found within a cell. So we have two categories of organelles, membrane-bound and non-membrane-bound. So the membrane-bound organelles are surrounded by a membrane, just like your cell membrane, that separates the organelle's contents from the cytosol, the liquid part of the cytoplasm. This makes sure that the activities of each of these organelles can do what it needs to do in sort of a relatively isolated and controlled environment. Each organelle has a specific shape, membrane composition, and its own specific enzymes that allow the chemical reactions to occur. So these differences make for the unique functions of each of these organelles. The membrane-bound organelles include the Golgi apparatus, the endoplasmic reticulum, lysosomes and peroxisomes, which are vesicles, and the mitochondria. Now we're going to look at each one of these individually, all of the organelles. The non-membrane-bound organelles are not enclosed by a membrane. They're either made of a protein by itself or a protein with RNA. They include the ribosomes, the cytoskeleton, centrosomes, and proteasomes. Now let's talk about these individually. So we'll start with the membrane-bound organelles, and starting with the endoplasmic reticulum. Most commonly we abbreviate it ER. So it's an extensive interconnected network of membrane that can vary in shape. So some parts of it look like little tubes, some of it look like sacs, some of it looks like flattened sacs. But the thing about it is there's one continuous lumen. So the lumen is the open space, okay? So one lumen within all of these little tubes and sacs. The external surface of the endoplasmic reticulum is a place where things can attach. So ribosomes and certain enzymes. We'll talk about the ribosomes in a few minutes. The endoplasmic reticulum that has ribosomes attached to it, as you see here in this image, all of this brown stuff, the little dots are the ribosomes. We call that the rough endoplasmic reticulum because it looks like cupcake sprinkles are attached to it, so it's rough on the surface. The smooth endoplasmic reticulum you see here does not have ribosomes attached, so its surface is smooth. Now, in addition to the endoplasmic reticulum, in this image, you see our membrane-bound organelles for the most part. There you've got your smooth and rough ER, your nucleus, your vesicles, peroxisomes, and again, we'll talk about each one of these as we continue. The ribosomes of the rough endoplasmic reticulum make proteins that will be released from the cell, incorporated into the cell membrane, and serve as digestive enzymes, or serve as digestive enzymes. The protein can be changed by adding other molecules like a sugar. So if we add a sugar to a protein, what do we make? Glycoprotein, exactly. Sometimes parts of the protein may be removed, like proinsulin. We have to remove part of the protein to turn it into insulin. But once these proteins have been modified, they're going to be packaged and stored until released. So they transport from the endoplasmic reticulum in small little membrane sacs like vesicles that pinch off the ER. These are called transport vessels. They transport the protein. They move the protein from the rough ER lumen, the inside, the opening, to the Golgi apparatus, which we'll look at next for further modification. So the amount of rough ER is a lot more abundant in cells that produce a lot amount of proteins for secretion, such as the cells in the pancreas that make insulin, and we use insulin, of course, to control blood sugar. The rough ER also helps to form peroxisomes, which are vesicles that contain chemical substances that we use to break things down. The smooth endoplasmic reticulum has a lot of processes, and that all varies based on the cell type. So the smooth ER can synthesize, which means to build, transport, or store different types of lipids. They can also be involved in carbohydrate metabolism, so glycogen synthesis and breakdown in the liver cells. The smooth ER is responsible for detoxification of drugs and alcohol and poisons. So you find a lot of smooth ER in things like the testes, because they make the steroid hormone testosterone, but also in the cells of the liver, because remember, the liver is your detoxifier that breaks down alcohol, drugs, and poisons. The Golgi apparatus is one of those with many names. Golgi apparatus, Golgi complex, Golgi body. It's all the same thing. It consists of about 5 or so elongated and flattened sacs made out of membrane. Those sacs are called cisternae. One of the primary functions of the Golgi apparatus is to modify, package, and sort proteins and the glycoproteins that we made in the rough ER. I think of the Golgi apparatus as like FedEx. It receives the proteins from the ER, modifies them as needed, and packages them up in transport vesicles and sends them off to do their job. In the cell, or by exocytosis, sends it out of the cell to go do its job. Lysosomes and peroxisomes. So lysosomes, lysis means loosening or breaking, soma means body. So these are small membrane enclosed spherical sacs that contain digestive enzymes immersed in an acidic fluid. So the fluid and the enzymes are inside the container. So the lysosome is the bag, okay? Now, lysosomes will digest unneeded or unwanted biological molecules. They can also help to get rid of damaged organelles. That process is called autophagy, to get rid of damaged organelles within a cell. When a cell is damaged or dies, the enzymes from the lysosomes are going to be released into the cytosol, the liquid within the cell. That is going to cause lysis or dissolution, breaking down. So that process is called autolysis or autolysis, if you prefer. So these little structures, the lysosomes, have 2 nicknames. They're called garbage men because they clean up and get rid of stuff that is unnecessary or unwanted, or suicide packs or packets because of their function in breaking down themselves when the cells are damaged. Peroxisomes are also small membrane-bound organelles that contain over 50 different enzymes that vary according to the cell type. Peroxisomes are also usually smaller than the lysosomes, but they're formed from vesicles that pinch off from the rough endoplasmic reticulum. Proteins are then added to the peroxisomes that serve as their enzymes. They engage in a lot of different metabolic functions. But you may be wondering, peroxisomes probably makes you think of peroxide, right? Well, they were first named based on their role in chemical digestion, which involves the removal of hydrogen ion from a molecule and produces a molecule of hydrogen peroxide. Molecules are broken down within peroxisomes, such as fatty acids, amino acids, uric acids, but not only do peroxisomes break things down, they also are responsible for some lipid synthesis or building of lipids. So your peroxisomes both digest, break things down, and synthesize or create molecules. Lysosomal storage diseases is a group of heritable diseases that are basically just characterized by the accumulation of not completely digested biological macromolecules within the lysosomes. It occurs because of mutations in the genes that code for one of the 40 different lysosomal enzymes. Tay-Sachs disease is one of those lysosomal storage diseases. So lysosomes in affected individuals, if they don't have that enzyme that's needed to break down membrane lipids, then those lipids will build up and accumulate within certain cells that have these complex membrane lipids, such as nerve cells, The cellular signs of Tay-Sachs are swollen lysosomes due to the accumulation of the lipid. Infants that are born with this typically appear normal at birth, but begin to show signs of the disease about 6 months of age, which can include paralysis, blindness, deafness, but they typically develop over a period of about 1 to 2 years, even though they may begin to show signs as early as 6 months. The progression of this typically is going to lead to death by the age of 4. There is no treatment. All right, just to review, the endomembrane system is an extensive array of membrane-bound structures that includes the smooth and rough endoplasmic reticulum, the Golgi apparatus, the vesicles, lysosomes, and peroxisomes. The cell or plasma membrane and the nuclear envelope are also considered to be a part of this endomembrane system. All of these structures are either directly attached to each other or connected through the vesicles that move between them. They're involved in various forms of metabolic processes that occur within the cell. They provide a way to transport molecules within the cell and release molecules from the cell. Substances are also brought into the cell as new vesicles are formed by endocytosis.